Hello, everyone. Thank you so much for your interest. My name is Ruth Flores. I'm a graduate student at Durango State University, just finishing a master's degree in molecular and cell biology. I'm here to present to you our work entitled Synthesis of a Biogenic Magnetite Nanoparticle KAEMPFEROL System for Biomedical Applications, a theoretical experimental study. Now I'm going to read to you the abstract. We present a study carried out by density functional theory, showing that the assembly of the processed magnetite nanoparticles KAEMPFEROL system is possible. Nanoparticles were synthesized using iron 2 and iron 3 salts as precursors in Laria-treated extract as a reducing stabilizer medium. Magnetite nanoparticles obtained have a particle size ranging from 7 nanometers to 15 nanometers when naked and up to 35 nanometers when covered by a silicon dioxide shell. To observe the effects of the system's components, alkaline electrophoresis was used for DNA extracted from MCF cells previously exposed to magnetite nanoparticles, magnetite core shell nanoparticles in KAEMPFEROL, proving that these cause DNA level damage in breast cancer cells inducing apoptosis. As a little introduction, we know that breast cancer is the second cause of death due to cancer in women. Available treatments are commonly expensive and cause severe side effects, but emphasize the need for new drugs that are efficient, harmless, and have lower costs. Magnetite nanoparticles have been proven to act as a vehicle for targeted drug delivery, increasing the therapeutic potential of commercial chemotherapeutics and bioactive compounds, as well as inducing oxidative stress target cells. KAEMPFEROL is a phytoestrogen with antioxidant and anti-cancer properties such as induction and metastasis inhibition. A magnetite nanoparticles KAEMPFEROL system is a possible drug precursor that might work as a treatment alternative against breast cancer because of the properties of its components. For the objectives, first we have to simulate a magnetite nanoparticles KAEMPFEROL system using DFT by means of quantum espresso. Then to synthesize magnetite nanoparticles by green methods and evaluate the effects of system components on a breast cancer cell line. And now for the materials and methods. For the theoretical methodology, density functional theory calculations were made with plane waves by means of the software Quantum Espresso. Such calculations allow us to determine how feasible it is to obtain a magnetite nanoparticles KAEMPFEROL system. Therefore, with the relaxation of the magnetite structure in bulk, LDIU pseudopotential was employed with a U of 4.5 electron volts and a polarized spin. The cubic structure of the 227 space group consisted of 56 atoms with octahedral sites for iron 2 and half of the iron 3, while the other half of the iron 3 was in tetrahedral sites. We worked with a ferromagnetic behavior and a 3x3x3 mesh. The relaxed structure was then taken into a surface lab on the 001 plane, which has had the most stability. Periodicity was carried with a 20 Armstrong vacuum. In the same cell, we added KAEMPFEROL to further relax, only this time we used gamma for K points under the BFGS method. For the experimental part, we used Laria-treated and dry leaves to make an aqueous extract in a 1:10 ratio. We then made an iron 2 and iron 3 solution in a 1:1 molar ratio, to which we added previously pulled down aqueous extract in a 2:3 ratio. Iron salts would act as a precursor for the nanoparticles, while the extract provided a reducing and stabilizer environment in which the needed reaction would take place. All of it was carried close in the flasks in order to prevent further oxidation of the product. These were washed with water, ethanol, and acetone until no remains of the extract were present. To add silicon dioxide core shell, we used Deos in alkaline environment as well as Aptus in water bath. The final product was characterized by means of transmission electron microscopy of CIMAV's National Nanotechnology Center. Now, for the testing part, we expose the MCF7 cancer cell line to the components of its system for 24 hours. For a visual inspection and an inverted microscope, we extracted the DNA in order to see if our nanoparticles of KAEMPFEROL cause any damage when inducing apoptosis. We observe this by using alkaline electrophoresis assay, which is able to show us DNA. And now for the results, as far as it goes, on the first picture showed on the very left, we can observe that it is in fact possible to have a magnetite KAEMPFEROL system, since we obtain an energy of 2047,145.85 Rydberg, in which we can see the KAEMPFEROL molecule embedded into the magnetite surface. On the other hand, the following pictures show us that when exposed to an strontium magnet, nanoparticles, both naked and core shell, exhibited a magnetic behavior. Micrographics show us that naked nanoparticles size range from 7 nanometers to 50 nanometers, while core shell nanoparticles had a size of up to 35 nanometers. These sizes are suitable and desirable for the intended biomedical application. Now, it is also important to mention that electrophoresis assay revealed that the system components we used in fact cause DNA damage on cancer cells, with a DNA degradation of up to 69% in contrast with the control lane, which can be seen as a bright white band with poor genome degradation on the left side of the image. And lastly, for conclusions, according to the calculations, it is possible to create an integrated magnetite KAEMPFEROL system. Nanoparticles indicate methods are used for application biomedical applications than that of conventional methods. Furthermore, components of the system have level on MCF7, meaning this could be a potential pharmacological. For the acknowledgements, we would like international of UHEC Faculty of Chemistry at London in 2021, nation from Lambda at UNAM, the academic body BFCE BIOGENIC in 2021, and CONACYT, Consejo Nacional de Ciencia y Tecnología 2020, for providing the necessary funding in order to carry out this research work. Thank you all for your interest and attention.